Hi, I'm Trey. This is Bailey. This is Tyler. This is Britton. And we looked at gut bacteria and how it mediates aggregation pheromones and German cockroaches. Hope we got a chance.
You have to put the arrows. Yeah, it doesn't, the touchscreen's not working, you have to put the arrow front and back and
Okay.
This piece's purpose was mainly focused on filling a gap of knowledge of how insect microbial presence affects organism communication. Specifically, this study dives into how gut bacteria affects aggregation habits. Volatile carboxylic acids, VCAs, are the main aggregation agents in this specific species of cockroaches. 40 VCAs are common in German cockroaches. The research realized while these VCAs were common in roaches due to fixation caused by bladybacterium, further research would help broaden their understanding of other insect species.
So first we're going to talk about a couple hypotheses that the researchers had. The first was that fecal VCAs that mediate aggregation in the German cockroach originated in gut bacteria, which are acquired from their external environment, and thus the insect's specific environment would shape its aggregation capabilities. The other hypothesis is that bacteria in the gut impacts VCA production, and VCA specific to each species cues the aggregation of members of that colony specifically. So German cockroaches were raised in sterile conditions which would lack certain VCAs necessary to promote aggregation. A simple reintroduction of those gut microbes in roach feces would lead to the restoration of regular aggregation tendencies. So just a couple more predictions. German cockroaches that use VCAs specific to their colony as a way to identify their own kin will be lost without the presence of those VCAs. And just some basics of the experiment. So we used first and fifth instar nymphs. We generated non-sterile feces and sterile feces. Identified bacteria in feces by using the PCR, which we'll talk about later, and observed aggregation behavior of each instar of cockroach.
So the lab strain of roaches that were used in this experiment were collected in Florida over 60 years ago. They used three groups. The control group, which 3 egg cases were separated and treated with 100 milligrams of feces from the original colony, and they were given food and water. The exenic group, which 3 egg cases were collected and sterilized and treated with 100 milligrams of autoclaved feces and given sterile food and water. And then the exenic bacteria inoculated groups, which were reared as exenic, but exposed to one or a mixture of bacterial isolates from the original colony. So for the bacteria to be counted and identified, the feces were treated with 10 milligrams and diluted 10 milligrams of feces was diluted in 10 milliliters of phosphate buffered saline. Colony forming units were counted and calculated per milligram of feces. And each morphologically different colony was isolated and identified by the amplification and sequencing of the 16S rRNA gene using 2 universal eubacterial primers, and identified using the basic local alignment search tool of GenBank. The test samples for the aggregation biopsies. So feces were collected from each group and extracted using sterile water, then vortexed and centrifuged. 2 synthetic mixtures were prepared, mix G and mix NCSU, and they were given to first and fifth instar nymphs with a choice of sterile water and control feces. So first and fifth instar nymphs were assayed in small petri dishes, separate petri dishes, and 2/10 of filter paper were treated with 15 microliters of the VCA or sterile water or control feces and placed on either side of the dish. 10 to 30 nymphs were introduced at one time and surveyed 24 hours later to assess distribution.
As for results, the results showed that gut microbiota contributes to the production of VCAs that act as fecal aggregation agents, and that cockroaches discriminate among the complex odors that emanate from a diverse microbial community. That's basically saying that the VCAs are different from each cockroach to cockroach community, and that they can, differentiate. Gut bacteria play a vital role in insect communication, and microbial communities present reflect the local environment. Therefore, fecal aggregation pheromones can be colony specific. We learned that of the first and fifth instar Blattodea nymphs, that first instar nymphs sheltered equally under 2 water-treated filter papers in a petri dish. This stimulus failed to garner much of a response from the older fifth instar nymphs, though. Both younger and older nymphal instars were attracted to fecal extracts from an adult female, preferring it over the sterile water. Water extracts of feces attracted fifth instar nymphs at a significantly higher rate, with them preferring the extracts far more than the sterile water. This shows the orientation responses of fifth instar nymphs to the synthetic mixtures of carboxylic acids that were viewed. The percent of tested nymphs that responded to both stimulus are shown on the graph. And these are just mix NCSU and mix G. Mix G stimulate nymphs to walk upwind in the olfactometer, but they had no real preference to this mixture. Mix G is composed of 6 compounds, but are presumably fecal aggregation agents of minimal to no importance. It should also be noted that the 3 compounds in mix G were not detected on the chromatograms. Mix NCSU, however, was made up of 6 major VCA compounds and was significantly preferred over all solvents and mixtures. In the largest doses, mix NCSU triggered up to a 90% response in all the roaches that were tested.
In the discussion, VCAs act as fecal aggregation agents that reflect the health, habitat, and diet of German cockroaches. Various semiochemicals are found in the feces, including aggregation agents, repellents, necromones, and food attractants. The gut bacteria influences the VCAs found in the cockroach feces and make them more attractive. Cophography promotes the transfer of VCAs. VCAs are produced by the cockroach and accelerated by the gut bacteria.
So the question we have for you is, why do you think the authors chose to focus on how VCAs affect aggregation semiochemical production when VCAs have also been linked to repellent and food attractant semiochemicals as well?